Human Genome Project is an international scientific research project with the goal of determining the sequence of chemical base pairs that make up human DNA, and of identifying or mapping all of the genes in our human genome. Our bioethical question is, must contractual consent be mandatory for a doctor to test a patient's genome, or for telling the patients about the results, even if they are in danger of a genetic disease?
The Human Genome Project started in 1987 when the United States Department of Energy wanted to study the effects of radiation on gene mutations. However, the conditions for the project were rooted as early as 1953 when Rosalind Franklin discovered photo 51 that revealed the double helical structure of DNA. Over the course of 35 years since the photo was taken, scientists were able to calculate the width of DNA, the 4 complementary base pairs, and how to sequence DNA base pairs. Specific DNA sequences were the most significant of these findings because it inspired the idea to decode the entire human genome. The Department of Energy wanted to work closely with the decryption of the genome to prevent mutations that resulted from overexposure to radiation. This government agency began the research in 1987 to decode an entire genome, when the National Institute of Health became equally interested in the exploration of this project. The NIH, another governmental corporation, honed in on specific sequences of DNA that were precursors to genetic information in order to provide medical information to patients preemptively. The decryption of DNA was extremely important to biologists that worked for the NIH, who focused their research on diseases that were inheritable, such as Alzheimer's and other kinds of cancer. The major agencies were funded by Congress to work together and make quicker progress of mapping an entire genome. They were under court order to share data and aid each other in the pursuit of the project. After nearly 20 years of hard, tedious work, the entire human genome was mapped by 2003. From a modern perspective, genetic information such as understanding predispositions for diseases and familial problems from the genome seems normal. Scientists, however, were only able to complete the mapping of the genome 13 years ago. Since then, people have had easy access to their genetic information, and the impact of this discovery has become commonplace. The main phase involved in the Genome Project was mapping, distinguishing genes that predispose humans to diseases and the SNPs related to them. The accomplishment of mapping the full human genome took years and is often trivialized. But before 2003, people were unable to access this information. The project was always controversial when it came to funding. People were often aggravated with the amount of taxpayers' money that went into its research. As costly as the mapping was, the U.S. economy has been able to reap the benefits of the completion of the Human Genome Project. The decoding of the human genome continues to be financially beneficial as it has brought almost $1 trillion to the U.S. economy. Overall, for every dollar put into the research, they got $178 back. Some economists argued over the projected financial improvement, but almost 15 years after the genome was mapped, it is clear that it has helped the economy. In 2013, the consensus was that the benefits were definitely on the higher side, close to $178, proving that the Human Genome Project was economically as well as medicinally advantageous.
What our genome really is the entire set of DNA or genetic instructions that our body contains. So every function that we perform, down to our cellular assembly of proteins, is regulated by this blueprint that we have that comes in the form of sequenced nitrogenous base pairs on a DNA molecule. We have 44 chromosomes that are made up of around 6 billion base pairs in total, and in the nucleus of each of our cells, this very long sequence is simply different strands of DNA wrapped tightly around histone proteins into a shape we call a supercoil. In terms of how a doctor actually begins to physically analyze your DNA, they first compare all of the SNPs present in your DNA with the completely dominant genome. SNPs, or single nucleotide polymorphisms, are small variations in a single nucleotide that occur at specific positions in your genome. To do this, they must complete a few major processes in the laboratory: harvesting cheek cells to work with from the patient, isolating them, exposing all of the DNA from their nuclei, amplifying a desired gene or section of DNA using PCR, digesting some of the DNA with a restriction enzyme, and finally running gel electrophoresis on the samples to actually determine the presence of an SNP. What you're seeing now is a time-lapsed running of gel electrophoresis.
There are several methods for acquiring genetic information from your genome. There is an algorithm that compares DNA sequences and DNA chain termination that is used when labs are sent samples of DNA to test. Nowadays, there are many companies, such as 23andMe, that will analyze your genome to determine genetic information for you. 23andMe has you send in a sample of saliva in a buffer that will preserve the DNA in a range of temperatures for transportation to their base lab. They will use different methods to analyze results and look for genetic predispositions. The algorithm used, called the Tiger Assembler, compares hundreds of thousands of base pairs in DNA to sift through normal sequences quickly. The Tiger Assembler guesses and calculates the number of base pairs in the genome using a combination of formulas and computer science. This new method for DNA sequencing was a necessary development because the old algorithm only compared a few hundred base pairs at a time. Labs will use the Tiger Assembler if they want an overall idea of what genetic predispositions are in your genome for statistics and further research. 23andMe also uses chain termination to analyze your genome. This method analyzes specific nucleotide polymorphisms, or SNPs, in your genes that you want to be tested for. Usually in labs, they will use this form of analysis if you have a hereditary disease in your family and are curious if it has been passed on to you. It does this by targeting a specific strand of DNA and replicating it with a polymerase chain reaction, and then uses restriction enzymes to cut the DNA at restriction sites. These sites are where there are SNPs that make each person unique and can also be precursors to hereditary diseases. If the DNA sample is cut, the scientists can interpret the results to either mean you have tested positively for a gene or you have the potential to express that gene. Then they will send you the results for you to process on your own.
The legal situation around the Human Genome Project brings another issue to the table: genetic safety. Around the United States, a general movement from the people has been made in the last decade. All over the country, there are new statutes and bills almost every year that are proposed to bring more security to people's genetic information and protect them from either genetic discrimination or exploitation of the info. Technically, your genetic information is your property. However, all of these bills, like the two in California and the three in New York, failed to deliver this protection, except one statute in New York. It states that prior informed consent is required to perform a genetic test. Further disclosure requires additional consent as well, and so we can see that the American people are conscious of and worried about their vulnerable and personal genetic information, though there is definitely a possibility that in the future companies, employers, and insurance agencies won't be so gentle with how they use our genetic information.
The Human Genome Project has the ability to prevent and cure diseases. It will also bring in around $45 billion for the U.S. It's beneficial both medically and economically. It can help diagnose familial diseases faster and cure the patient earlier before too many symptoms occur. Other people argue that the Human Genome Project is useless and that we should be focusing on our efforts on preventing the diseases from spreading through the environment. The project costs too much and does not benefit society because it doesn't create any new breakthroughs. Scientists can only prevent so much on a microbiological level because the environment affects people so much. It's impossible to prevent diseases just by looking at the patient's genome without looking at the possible effects the environment had on them throughout their life. Even cures can cause worse health effects. Iatrogenic diseases, which can sometimes occur when drugs are used to try to cure a disease, can sometimes be worse than the disease itself. The Human Genome Project can also provide the U.S. government lots of citizens' information. Having access to their genomes reveals very personal information about the subject. It's very hard to escape the surveillance of the government when they have so much information about a person through their genome. If a company which stores genetic information receives a request for someone's genome, there is no law stopping them from giving it to them. Anyone can have access to a person's genome and therefore personal information. It is dangerous to have that information so available to the public. Companies want to make genomes accessible to the public. Dr. Church, a scientist who studies genomes, encourages everyone to put their genomes online, available to anyone, anywhere, to be able to compare and contrast genomes with other people. Many people have been enthusiastic about the Perspective project, eagerly adding their genomes to the database. This database, holding many genomes, which expresses many traits about each person, is then viewed by different types of scientists, trying to make sense of the genomes from all different perspectives. Anyone can have access to the database and each genome and possible diseases of that genome's owner. The database is meant for people to know if they're susceptible to a disease by looking at other genomes where the disease did occur. For example, Dr. Church added his genome to the database, and a doctor who was also on the site noticed that he was not treating his cholesterol correctly. Dr. Church then took a test and found out that the doctor online was correct, and he changed his medication, which ended up improving his health. The people who are adding their genomes do not have to attach their name to the traits. This allowance helps protect people who could get a disabling disease in the future from being discriminated against by employers. Ultimately, we chose to be for the Human Genome Project because it helps cure more diseases than it causes. It also helps prevent many hereditary diseases while simultaneously being economically beneficial.
You're as good as dead.